Today is April 25th, 2017. My name is Megan Mitzelfelt, and I am at the Chicago McCormack Place Convention Center during the annual meeting of the American Physiological Society. Today I have the pleasure and the honor of interviewing Dr. Charles Pat McCormack for the Society's Living History Project. Dr. McCormack is a long-standing member of ApS who's been affiliated with the Chicago Medical School since 1964. Dr. McCormack's research has focused on reproductive endocrinology and circadian characteristics of the LH rhythm. Pat, welcome to the Living History Project, and thank you for agreeing to be interviewed for this project. If you are ready, I'd like to ask you a few questions about your life and your science.
That's fine, and I'd like to thank you and the Society for giving me this honor.
Yes, of course. Okay, so Pat, you were born in rural Illinois and then moved to a farm in rural Wisconsin. Can you tell us a little bit about your family, receiving your education in a one-room schoolhouse, and then the role the farm played in your interest in science?
Well, my grandfather and my father were farmers, so naturally I thought everybody was a farmer, and it was great growing up with my older sister, my older brother, and then later three younger ones. Always someone to play with, always someone to do things with, and on the farm you had a lot of opportunities for trying things. It was great, and the one-room schoolhouses are interesting. They accomplished much of what the educators would like to have today, namely personal attention. You couldn't get away without answering the teacher. You had your classmates there who were listening to you, so you had an immediate audience. And the largest classroom I ever was in would be 25 students, and oftentimes 6, 7, 8. So that was very good education in retrospect.
Mm-hmm. Yeah, I'd imagine so. How did that pique your interest in science?
Interest in science came kind of slowly. One of my early Sunday school teachers mentioned to my mother that this guy keeps asking questions. He doesn't believe what I tell him. And my first grade teacher said to my mother and father, "I'm going to nickname him Y."
That's
because
funny.
Whenever she would tell me something, I would ask why. Whether it was pertinent or not, I was asking why. So I think that, I guess I'd have to claim some genetic input into that, though my father and mother were both inquisitive people as well, so I lucked out.
It was nature and nurture.
I would think so,
Yeah.
Yes, thank you.
Okay. So in high school, you developed an interest in biology and chemistry, and you planned to go to college to study to become a medical doctor. Can you tell us how your years at Carroll College contributed to your career decision to move into research and a graduate degree?
Well, again, there, I was in small classes. I had two fantastic professors, Dr. Nance and Dr. Christoph. They made biology very interesting. The chemistry department made chemistry very interesting, so the two dovetailed together very nicely. High school kind of led up to that. I had good classes there too, and my performance was okay, was not exceptional, but fortunately it worked out, and I did do a little bit of research in college over one summer. I collected flies from fly traps that were common at that time, and the interesting thing is these traps were designed to catch houseflies because houseflies bothered people a lot in August. And the conclusion of my research, because I collected these flies over many weeks, is that the one thing they didn't catch was houseflies. They captured a lot of very helpful dipterans, flies that ate dead animals and ate feces and so forth, kept the cycle, the organic cycle going, they probably were counterproductive in many respects. I didn't realize that at the time, but at least I had the sense to make the conclusion that they're not catching houseflies.
Yeah. Is that what led you into wanting to go to graduate school?
Well, what really led me into graduate school, I was pre-med and doing okay, and I'm quite sure I could have gotten into medical school, but one day I looked at the bulletin board, and there were these flyers about graduate biology degrees, and I asked my professor, "What is this all about?" "Well, you go to school and you take some classes and you do some research, and how do you pay for this?" I said, "Oh no." He said, in graduate school you don't have to pay tuition. In fact, they'll give you a little stipend. Thus ended my medical career right then, because I knew that was what I really wanted to do. And if someone would pay you for doing research, what more could you ask for?
I felt exactly the same way. That was pretty much what led me to it. Okay. So for your graduate degree, you ventured to the University of Wisconsin-Madison, where you received your PhD with Roland Meyer in the endocrinology training program. Can you tell us about your doctoral research, what you did there, and what you discovered?
Well, Dr. Meyer had a dozen graduate students. He couldn't keep track of all of them at all the time, so they helped one another a great deal. And he liked to get you started on research right away. Within 2 weeks after I was there, I was doing experiments. I didn't know what I was doing, but I was doing experiments. And he put me on a project which was quite interesting. At that time, they knew that the brain, the hypothalamus of the brain, was controlling the release of hormones that caused ovulation. They knew that and were trying to isolate that hormone, so he was in the fray of that, trying to localize and isolate that hormone. So I was on that project, taking hypothalami from pigs, grinding them up, injecting them into immature rats whose ovaries had been stimulated with an FSH compound, follicle stimulating compound, and seeing if I could get them to ovulate. Well, I couldn't. Of course, if I gave chorionic gonadotropin, which is a luteinizing hormone compound, that did work, but these hypothalamic injections did not make ovulation occur. So Dr. Meyer said, see if you can sensitize the animals. The way you're supposed to sensitize animals to LH is by giving estrogen. So we gave estrogen, and when we gave estrogen, what we did is instead of none of the control animals ovulating, about 10 or 20 or 30% of the controls would ovulate, but still the hypothalamic implants, injections did not cause ovulation. So obviously we didn't have the active stuff in our hypothalamic extracts. But I tried one more sensitization technique. Everett and Sawyer had shown maybe 7 or 8 years later that progesterone could cause adult female rats to ovulate a whole day earlier if given at just the right time. So I thought, well, I'll give a little progesterone, not enough to make my immature rats ovulate, release LH and ovulate, and that'll make them very sensitive. So if I have a little bit of the LH releasing hormone from the hypothalamus in my solution, injecting it intravenously, I'll get those little rats to ovulate. I chose a dose of progesterone that Everett and Sawyer used, an entire milligram, and to my surprise, all of the control rats, those that were given just isotonic saline rather than the hypothalamic extracts, ovulated. So that in itself was very interesting. I had found a way, inadvertently, of studying this progesterone facilitation of the LH secretion. Up until that time, most everyone thought that progesterone was an inhibitor of LH secretion, and even those who knew about Everett and Sawyer's study thought, well, their progesterone was working by virtue of storing up some luteinizing hormone in the rat's pituitary so that more was there to be released. So nobody took the notion that progesterone would actually cause LH secretion seriously. I believe my studies, that went on to become my entire thesis study, contributed greatly to the notion that progesterone at a certain time can strongly facilitate the luteinizing hormone secretion in animals. The same has been shown now in monkeys and people, that progesterone at a certain time can strongly stimulate the secretion of luteinizing hormones. So that kind of put me in business and made a PhD thesis for me and allowed me to get my first job.
Yeah. So your first job, let's talk a little bit about that. Where did you go? What did you start researching then? Was it different from your graduate school research, or?
No, the reason I took a job in Chicago was twofold. First of all, tragically, 1 year earlier, my wife, who was from Chicago, her parents were killed in an automobile accident. At the same moment they were killed. And she had to go to Chicago to settle her family's estate. They had a 2-flat building in Chicago that had to be settled. So it was kind of imperative that I go to Chicago. And there was this job in Chicago at Chicago Medical School, where I could continue my research. The teaching load was not terribly heavy, and there would be time and opportunity to do research. So I took the job, in spite of the fact that it was the lowest paying job offer that I had. I took it because we had family business to take care of. Well, I'm still there 54 years later, and it turned out to be a wonderful position.
Great. So did you take your graduate work with you? Did you continue on that path? Did you change it in any way?
I did continue that project. I had some more ideas that I wanted to do on the progesterone facilitation of the luteinizing hormone surge that caused ovulation. But I also, in the last year or two of my graduate training, a friend of mine, very good friend, Bill Strauss, was doing a project also in rats where he had shown that, again, in immature rats, that you can control the time of the luteinizing hormone surge and control the time of ovulation by changing the light-dark cycle. And that really got me into circadian rhythms, which intrigued me. And I did a little bit more work on the progesterone facilitation of the LH surge, but did a whole lot more work on studying the circadian rhythm that controlled the timing of ovulation in rats. That pretty much occupied my interest for the next 25 years. I did a little more on progesterone, still would like to do more yet, but I got into circadian rhythms in a big way. And I think that worked out well. I did well and got promoted and got some grants, and it was a lot of fun.
Okay. Okay, let me see. So in 1988, after losing extramural research funding, Richard Hawkins, the department chair, asked you to become director of the medical physiology course. So can you share your thoughts about this request and how you used the experience to become a well-recognized teacher in physiology?
Well, let me elaborate on that just a bit.
Okay.
I had a grant, and it was going okay. I was doing good things. But I was very intrigued by the teaching. Also, I was doing some national board review teaching to earn a few extra dollars. And that was going fine, too. So when Dr. Hawkins, who parenthetically is still my good friend, asked me to do this, I said to myself, you don't really have any choice here, McCormack. You don't have any money, and you better get going on something. So I took it. And that, along with this extracurricular teaching that I was doing, forced me to learn all of physiology. I had to read the classic textbooks, prepare lectures in many different areas. So in some respects, it was a fortuitous change, and I got into teaching. Whenever we would lose a lecturer in a particular area in our department, I would fill in and I'd lecture cardiovascular, respiration, gastrointestinal. So I ended up being an all-around systemic physiologist, which works well when you're running an entire physiology course. You can monitor the questions of other professors, help to write better exams, and that worked out well, and I'm happy to say that the students seemed to appreciate it.
Yeah.
Gave me a few golden apples, and that worked well for me.
Wonderful. Okay, so as you reflect on your research career as a reproductive endocrinologist, what would you consider to be your most significant contribution?
I reflected on that a few weeks ago, and indeed, what I did in graduate school, showing that progesterone could be a very powerful stimulator of luteinizing hormone secretion, has been my major contribution. And I still hope to follow that through in the next few years, write some review articles on that, and try to encourage some others to get back into that work. With respect to human ovulation, I believe that with progesterone administration, which is a natural hormone, given at the right time in small doses, we could get women to ovulate every 28 days precisely. Now, my granddaughters interestingly say, "Grandpa, why would you want to do that?" and I'm not sure I can give a good answer to that because I'm not a woman. But I believe there's a significant number of women in the United States who don't like oral contraceptive pills for one reason or another. And if they were using rhythm control, it would be nice to know exactly when they were ovulating to prevent conception. Also, if you could count on ovulation at a particular time, it would enhance your capacity to become pregnant if and when you decided that was the time for you. So I think there's a lot of potential for that yet, and I hope to exploit that a bit in the next few years.
Certainly, and you should, especially with the growth in reproductive assistance.
Yes,
Yeah.
I think it is. Well, the world population is going like crazy, but interestingly, in the United States, amongst professional women like yourself, having children gets to be a problem because by the time you get a meaningful profession, a meaningful job, you're oftentimes in your early to mid-30s, and that's the exact age when fertility starts to decrease.
Right.
So I think I have a few more things to do yet.
You do, certainly. Okay, so can you tell us a little bit about your first ApS meeting and how it differs now from this meeting that you've been to?
Oh yes, I was talking to a graduate student on the bus coming in from Chicago this morning about that exact topic. The first federation, I think at that time they're called federation meetings. Now we call them, it seems to now experimental biology meetings. The first meetings I attended were in Atlantic City, New Jersey. Year after year, they'd have the meetings in Atlantic City, New Jersey. There were a lot of hotels there, and you could get a hotel room for $30 or less a night, which was a big difference from now. And that's where the meetings were. So lots of things going on, and that was the heyday, so to speak, of neuroendocrinology. Lots of great stuff was going on. Really top-notch people were there reporting their research. Ernst Nobel, Nina Schwartz, Joseph Mites. These names will mean something to other people. And it was so wonderful getting to know these people in the field, and occasionally one would say, "Oh yeah, you're the guy that did that," and that gave me a great deal of good feeling for myself that I'd done something that these top people in the field had noticed.
Mm-hmm.
So I must say that's nice.
So how are they different than the current meetings?
Oh, these meetings are so huge.
Yeah.
These are so huge. There's always something to do. I was at meetings this morning from 8 o'clock until 10. There's always something going on, many different areas. In those days, there was almost no research on humans. It was all experimental biology in animals. Mm-hmm. Now there's a lot more studies being done on humans because we realized that the things we learned in animals have applied very much to people. So a lot of that, very practical things, how to prevent obesity, which impacts greatly on fertility, prevents heart disease by preventing obesity, preventing diabetes mellitus. The society, at least the nutritionists, the physiologist, and I suspect the biochemists too have gotten into this a great deal. So it's a much broader meeting. I think the early meetings I went to might have had 2,000 or 3,000 attendants, and now I'm sure we're over 20,000.
Yeah.
It's amazing.
It is huge growth, right?
Yeah, you used to be able to get all the abstracts in a thin little book, a few, maybe 400 or 500 abstracts, all in a thin little book. Now they don't even print the abstracts. You have to use your little handheld computer to find a person's abstract.
Okay, so tell us about teaching physiology in the medical curriculum today and how that's different than it was taught earlier in your career.
Well, medical education is undergoing a marked change at the present time. Originally, physiology included all of neuroscience. When I joined our department in 1964, in fact, I first lectured in neuroscience. Again, that was a good thing and broadened my horizons. But we taught everything as courses. Everything in medical school was a separate course. Over the years, we've merged courses, and now the typical, up until a couple of years ago, the typical curriculum was where the knowledge, medical knowledge, was organized around the systems: cardiovascular system, respiratory system, gastrointestinal system. That was the typical physiology course. Now the movement is to organize medical knowledge around conditions: heart disease, lung disease, gastrointestinal gut diseases, as they call them, pathologic conditions, and physiology, the biochemistry, all of it put together into a unit, organized around the condition or the disease. And that is supposed to make it easier for students to put the knowledge together, understand it, and retain it. That's the notion. We'll find out whether it's going to work or not, but that's what a number of schools are turning to, and that's interesting. The Chicago Medical School, where I teach, will begin that kind of a curriculum in August of 2018, and I look forward to being a small part of it.
Mm-hmm. So in your opinion, is that beneficial? Is that a good change that we're going towards that? Or do you think the old way was...
Well, I think it is a fine way of doing it. The problem is that the instructors need to know more than they need to know when you only taught your own small little part. You have to have a bigger, broader knowledge of the cardiovascular system to give your physiology lectures, bigger, broader knowledge of the endocrine system to give your endocrine physiology lectures. I hope that the physiologists will be able to adapt to this. If not, I suspect much of the first 2 years will be taught by clinicians rather than basic scientists, if physiologists can't broaden their horizons to do that. We'll see.
Okay. So what role has the APS or other scientific societies played in your career?
Well, the Physiological Society has been very good to me. I remember on a couple of occasions when our department was struggling a little bit, Physiology, American Physiological Society sent out experts and consulted with our department and tried to help us get back on track. I think that was very nice. Also, about midway in my career, while I was still doing research, I heard about the Porter Foundation for supporting minority students. In our department at that time, I was acting chair, and we did have a minority student who needed support. And the Porter Foundation was able to support her, was willing to support her. She turned out she did a very fine project on conduction of the stimulus that causes the heart to contract. She did a very nice project and went on to have a pretty decent research career. So they were involved in that. Marty Frank has been good to our department over many years. I've gone to several of the special meetings that Physiology has had over the years on exercise, metabolism. Those have been great. It's been a very fine society, and I believe today it's still very supportive to the young people especially. Yes. I noticed each year they have a meeting where the local high school and undergraduate students interested in physiology present posters and do just a terrific job on that. So yes. Very helpful in many respects.
Wonderful. So what advice would you give to students who are starting out in their research career or in science today?
My first advice would be sort of a social, psychological thing, and that is, don't allow yourself to be overwhelmed. Those men and women who are up there giving the session lectures, the summary lectures, are no smarter than you. They're no more capable than you. They started out just like you, and amazingly, things fell into place, and now we look at them as if they are really geniuses and extremely productive people. Well, they were just graduate students like you. And if you put your mind to it, and if you're doing something that you enjoy, you'll get there. You'll get there. And also, don't be too alarmed about what you hear on the news, that all the money is going to be cut off for research, and there's not going to be enough support any longer, and schools are going to close down and all this. In my 54 years in this teaching research business, I must have heard that a half a dozen times. And the societies are bigger and stronger, and the research they're doing is better than it was many years ago. There's a place for you as a student. There's a place for you. You have to be adjustable. You have to be willing to take opportunities when they arise. You may have to change your research area now and then. But if you love research, if you love knowledge, it's a good place to be, and physiology is a terrific part of that whole business of finding out what nature is all about.
Right. Well, thank you very much. It's been a pleasure interviewing you, Pat.
It's been my pleasure talking to you, Megan.
Yes.
And I appreciate the opportunity to do this.
Yes.
Thank you.
Thank you.